Hello everyone, welcome back to Ravi's biology classes. Yesterday only we have discussed about the excretion topic, and we have briefly understood yesterday that how animals are classified on the basis of the excretory products they secrete, and also we have seen the excretory organs of different invertebrates. Finally, as I have told that today we will discuss about the excretory system in human beings. I wish that you all will enjoy this session, learn about the excretory system in human beings. As we see in the excretory system, this forms a organ system of the human body. So, organisms, rather animals, have advanced in their complexities. Organ systems have also developed, and excretory organ is one such organ system. As we see in all other organ systems, excretory system also comprises of set of a few very important organs. So, here we find that human beings and all the mammals, they have two kidneys. So, in the case we can say that a pair of kidneys are present in human beings. Kidneys are present in lumbar region. If ever you study about the skeletal system, and we study about the vertebral column, and there are 5 vertebrae. So, between these 5 vertebrae, in the region of these 5 vertebrae which are labeled as L1 to L5, the human kidneys are present. So, how many kidneys are present? They are 2 in number, means they are 1 in pair. Human kidneys are bean shaped and reddish brown in color, and the blood vessels which supply blood to kidneys are called as renal arteries, and the blood vessel which takes away the blood from the kidneys is called as renal vein. Kidneys are the site where the urine is formed. The urine which is formed is transported to a temporary storage organ with the help of a long tube like structure called as ureter, and it gets stored in a bladder like structure which is called as urinary bladder, and finally the urethral opening allows urine to be released out. So, in a nutshell, we have just understood about the human excretory system. So, let us see what we observe when we take a lengthwise section of the human kidney. In the lengthwise section of the human kidney, that is generally called as LS of human kidney, longitudinal section of human kidney. We observe that our human kidney has two distinct layers. The outer layer, which is lightly stained, is called as cortex, whereas the inner portion, which is darkly stained, is called as the medulla. In other terms, you can even say it is also called as renal medulla. Renal medulla is further divided into 8 renal pyramids, which has the broad base towards the cortex and the conical portion towards the center. So, these are called as the renal pyramids. Each renal pyramid contains a set of structural and functional units of human kidney that you very well know what they are. They are the nephrons. And if you notice the how many nephrons are present in each kidney, they are 1 million, or we can say that 10 lakh per kidney, right? Okay. We will discuss about the structure of nephron and the mechanism of urine formation in detail just after a moment. These renal pyramids, they converge towards the center, as I have told you earlier, and the central position is called as renal pelvis, which actually opens up into a tube like structure which is called as ureter. So, what does ureter do? Ureter takes away the urine to store in urinary bladder. Here the blood vessels are also mentioned. The red color blood vessel is called as renal artery and the blue colored blood vessel is called as renal vein. Thus, we have just observed the longitudinal section of human kidney, and now we are going to discuss about structural unit of the human kidney, that is a nephron. Also, we will understand the mechanism of urine formation in the nephrons. So, the nephrons are the structural and functional units of the human kidney. It is a highly coiled structure which has a lot of tubes running in a very coiled manner, and also the blood capillaries surround it so that the absorption of the useful materials take place effectively. So, let me help you to understand the process, the structure of the nephron in a simplified manner. What do we find here? Nephrons are having a globular structure which is a cup like structure. This cup like structure which is present is called as Bowman's capsule. This Bowman's capsule receives the blood from the renal artery, and though renal artery, when it enters into the kidney, it divides into two arterioles. The first one is called as Afferent Arteriole and the second one is called as Efferent Arteriole. Now, the difference between both of them is Afferent Arteriole brings the blood to the nephron, and the blood goes into the Bowman's capsule through the Afferent Arteriole, and Efferent Arteriole takes away the blood out of the nephrons, out of the Bowman's capsule. Once the Afferent Arteriole enters the Bowman's capsule, it divides itself into a tuft of blood capillaries. You can see the tuft of blood capillaries are present here, and these complicated blood capillaries network which is present here is called as glomerulus. So, let us understand one basic concept over here. When an artery which is thick, it divides itself into branches, it becomes thinner, and when it further branches into even finer blood capillaries. So, what we understand about the blood pressure running in the or experienced inside these blood capillaries, the blood pressure will be less, but as the artery becomes thinner and thinner, the blood pressure, that is blood pressure, it starts to increase, and in the capillaries the blood pressure is quite high. So, the blood flows at a very high pressure when they reach in the glomerulus, when they form the glomerulus. So, what we understand, the blood which is entering through the afferent arteriole, it is under huge pressure as it enters the Bowman's capsule and forms the glomerulus. One more thing to understand here is, what is the nature of the blood that is received by the Bowman's capsule? Blood which is received by the Bowman's capsule, it is rich in wastes. And what kind of wastes are produced in the human body? They are primarily urea. Apart from that, we have to understand one more thing, or rather you can say 20 to 25% of cardiac output. Cardiac output means the amount of blood which is pumped out from the heart, and out of that 20 to 25% is carried by the renal artery, by the afferent arteriole into the Bowman's capsule. So, the blood is under huge pressure when it comes into the glomerulus. Filtration takes place in the glomerulus, and as the blood is under huge pressure, the filtration will also be extremely rapid, and thus this is called as ultrafiltration of blood. When the ultrafiltration of the blood takes place, it results in removal of many of the unwanted wastes, and wastes are urea, bile salts, dead cells. But apart from that, some useful substances also get removed. As you know that blood is composed of water. It also contains lot of mineral ions like sodium, chlorine, potassium. Also, if the blood contains glucose, so glucose will be removed. Thus, what we see that ultrafiltration results in the removal of wastes, and also it results in the removal of the useful components, right. That is why this all has happened just because the high pressure of the blood that goes into the glomerulus. Now, what is the function of the nephrons? Nephrons have to remove the waste, but they have to also reabsorb the useful components back into the blood. So, let us now proceed further. The filtrate, after going through the ultrafiltration, it moves into a coiled tube, and this coiled tube is called as proximal convoluted tubule. So, this particular portion is called as proximal convoluted tubule. In a short form, it is called as PCT. As the filtrate runs down the proximal convoluted tubule, what is going to happen is reabsorption. What do we mean by reabsorption? It means absorption of important molecules or the components from the filtrate back into the blood. So, as we have seen, what are the important components of the filtrate? Water, sodium, potassium, chlorine ions, and some amount of glucose if it is present inside the filtrate. They have to be brought back into the blood so that we do not suffer from any kind of dehydration or loss of the nutrients or mineral ions, right. Now, how it will take place? If we observe the afferent arteriole, once it enters into the glomerulus, it also branches out and forms a tuft of blood capillaries which runs around the proximal convoluted tubule, and the complete tubules of these, and they form a tuft of blood capillary network around these tubules. And the tuft of blood capillaries which run around these tubules, they are called as vasa recta. And the function of the vasa recta is to reabsorb the excess amount and the useful components back into the body fluids. So, they are called as vasa recta. At the same time, we have to consider one more thing, that the proximal convoluted tubule, it is thicker. So, the reabsorption process, though it will start in the proximal convoluted tubule, but it will not be so rapid. And one more thing, the PCT lies in the cortical region of the kidney. So, I have tried to make a separation between with this black line. So, this black line, that above it is called as the cortex, and below it is called as the medulla. So, PCT is lying in the cortical region, and the thickness of the PCT does not allow high amount of reabsorption to take place. Okay. And as the PCT runs down, it becomes thinner. As you can see in the diagram, the PCT becomes thinner, and this thin tubule which runs runs long and down into the kidney, and which region it is? This is called as the medulla region, and this long thin tubule which runs down into the medulla region is called as loop of Henle. One more concept has to be remembered over here, that as the depth increases, the atmospheric pressure increases. Over here, osmolality increases. So, what happens, as the tubule is becoming thin, the depth is increasing into the medulla. As the tube is running into the medulla, the reabsorption rate also increase. So, this loop of Henle, as I have told you, it is running down. So, what it is termed as? This is termed as descending limb of loop of Henle. And similarly, this particular region is called as the limb which is rising up, it is called as ascending limb of loop of Henle. So, in both these tubules which run down and up in the loop of Henle, the reabsorption rate increases many folds, and the filtrate which runs into these tubules gradually becomes concentrated. As the filtrate runs into the thick tubule after going up through the ascending limb of loop of Henle, the thick tubule which is shown in the green is called as distal convoluted tubule. In short form, it is called as DCT. So, the filtrate which runs into the DCT is actually highly concentrated, and it is having less amount of water, and all the waste products are present in the filtrate, and now the filtrate is called as urine. The filtrate present in this DCT is called urine, and finally the filtrate is drained into this thick tubule, and this tubule or this duct is called as collecting duct. If there are, so there will be n number of collecting ducts. So, there will be as many collecting ducts as many nephrons will be there, and finally the collecting duct joins with ureter, and after that you know that ureter joins with or stores the urine in the urinary bladder, and from the bladder it is excreted out through urethra. So, in this manner we have seen that how a nephron purifies the blood and forms the urine. The two major steps which are included in the formation of the urine are the ultrafiltration of the blood and the reabsorption. So, students, in this video we have discussed about excretory system in human beings. Then we have seen that how kidneys look under the longitudinal section. Also, then we have finally discussed in detail about the structure of nephron and the mechanism of urine formation in nephrons in human beings. So, if you have understood this topic, or if you have any kind of confusion, if any kind of queries are there, you can post me in the comment boxes. And if you have understood this topic nicely, then please like and subscribe the channel. But it is even more important that you comment in the comment box that how did you like this content explanation, and what improvements can I make to make the content even more effective for you to understand. Thank you.